last class we were talking something about the requirement of an RNA as an intermediate when the genetic information has to be relayed and being executed as the protein information or being converted into protein information, whether there is a need of having an RNA intermediate. That was what was the question that we were looking at in the last class. So if and I gave you the time of entire week or so in order to think over it. So if you have come up with some answers, then let us start discussing in that direction first, and thereafter we will find out certain ways in which a proof was established in order to confirm the existence of an intermediate molecule which is going to relay the information that is there in the DNA to execute the information in the form of proteins. So have you come up with some answers, or did you find some answers to the basic question that was asked? Should I repeat the question once again for you all? Okay. I hope that you remember the question. The question was regarding existence of RNA, or rather requirement of RNA as an intermediate molecule that is going to be used for relaying the information that is there in the DNA to the executioner of the information, that is protein. So the question was simple, that if the information is there in the DNA, then why can't DNA directly be converted into protein? Why do you require an unstable intermediate like an RNA molecule? So has anybody arrived at any answers to this question? Partly one answer was already given in the last class itself, but we need to discuss more upon these lines so that we can establish a very solid grounds that why there is a requirement of an mRNA molecule to relay the information from DNA to protein. Okay, so does have any one of you paid any attention in trying to find out the answer for this question? Yes?
Sir, I have a few guesses.
Okay, Bhavika, go ahead.
DNA is because DNA is inside the nucleus and protein production machinery is in the cytoplasm. Hence, we needed something to relay the information from nucleus to cytoplasm for protein production.
Yes, in one way that is correct, but other way around, if you look at it, in case of bacteria, uh, all the two processes happen simultaneously, right? Transcription is immediately followed by translation. So there is no nuclear envelope there, which is serving as a hurdle for the information. And presently, although we are talking about bacteria, uh, but obviously we need to have a solid proof for all sorts of organisms that we are referring to. So Sarvesh has something to say. Proteins are translated on ribosomes, which are present in the cytoplasm, and so DNA would have to migrate. It is more or less on the same lines, Bhavika, what you were trying to say. So So, okay, fine. I can take that as one of the reasons why do you require a messenger RNA molecule, perhaps to cross the information from one chamber of the cell or one compartment of the cell, which is nucleus that harbors the DNA, to another compartment of the cell where the protein machinery is located. You need that message to be relayed from one compartment to other. Okay, fine. That tells us something about the requirement of a molecule in between. But as I said, in case of bacteria, this happens simultaneously. As soon as the mRNA starts to emerge out from the transcriptional machinery, it is immediately captured by the translational machinery, and the translation process begins. And this is a very important reason why bacteria have a different kind of transcription regulation system. And you'll know this, or you will learn this when we go into regulation of transcription in bacterial cells. Okay, Bhavika, go ahead for your the second guess.
It is like that one mRNA, due to splicing at different sites, may give rise to a set of proteins. But if one
Again,
gene,
you're talking about eukaryotic system, right? You're not talking about the bacterial system. Why I ruled out your first reasoning was because in case of bacteria, you don't have that compartmentalization, right? So in case of bacteria, immediately you can translate the mRNA that is being coming out from the transcription machinery, correct? So there is
yes.
no splicing happening in case of bacteria, correct?
Oh, I didn't know that.
So so in case of bacteria, yeah, in case of bacteria, you can directly translate immediately as soon as the mRNA is getting transcribed. So in case of bacteria, transcription and translation are simultaneous processes. As opposed from in case of eukaryotes, transcription and translation are two different processes, right? And you need processing of RNA to happen in between. So in eukaryotes, the thing holds good. But in case of prokaryotes, where you are actually discovering the process, does that seem to be an important requirement that you should have a messenger RNA in between? Does that make sense? Okay, Sarvesh has something to say. In DNA, there are a lot of non-coding regions, so it will take a lot of time to search, and it will also be inefficient energetically. I can convince you, Sarvesh, on this point that it will be energetically more favorable if you use DNA directly to serve uh, as the information for RNA. For the protein synthesis. I can convince you on this point. Imagine now. Okay. Okay. So imagine now that this is the gene that we are talking about, just for simplicity's sake. Okay. mRNA is essentially this same sequence, right? The sequence which is shown in green, blue here in the gene, it's essentially the same sequence which is in mRNA, except that the T is getting replaced with U in mRNA, correct? Now imagine this, that I am able to read this sequence directly and convert it into protein instead of going through mRNA in between. Will it not be energetically favorable? Because for every base that is going to be utilized for synthesis of mRNA, you are going to spend 2 ATPs for addition of every base. So it's energetically more favorable considering the gene directly being utilized for protein synthesis. Do you agree with me now? And about the non-coding part, the non-coding region is only again in the coding sequence, the non-coding region, the part of non-coding region that you're talking about is essentially present only in case of eukaryotes. Those are introns, they are called as introns, and exons are coding, correct? But that is again a eukaryotic situation. We are not essentially referring to eukaryotes at this point of time. We are referring to prokaryotes. So any other reasoning that you can give? Yes. Are you finding my defense logically fitting to the reasonings that you are giving? Yes, Bhavika?
I couldn't hear your question, sir.
Okay. Is my voice breaking or anything like that?
No, sir, there is problem with my network.
Okay, fine. So I will repeat the question again for you. Are you finding the reasoning that I'm giving for the defense that I'm putting for in order to disregard the explanations that you are giving? Are you finding that defense logically good enough or not?
I mean, why would a system want to waste more energy?
Exactly. So that's what my basic question is, correct? Why would a system want to waste excess of energy? So you should have a proper reasoning why you are expending more energy in synthesis of RNA.
Sir, so just an example, like in embryonic development, from there onwards, only
Hmm.
we start the protein machinery, protein producing machinery. So during that, when cells are being differentiated into different kinds of cells, some of the part of their DNA is completely stopped from getting transcripted and translated. So if
Right,
mRNAs were
because
not
the
there,
entire so again you are talking about the eukaryotes, right? If you're talking about embryos, you are again referring to eukaryotes. Eukaryotes is a complex system. Had it not been important, it would not exist in prokaryotes as well, right? In case of eukaryotes, yes, because of complexity, you need this to be present, right? Am I making sense?
yes, sir.
Okay, in case of prokaryotes, it's a simple system, correct? In that also, why you need mRNA? That's the question that you need to address, Bhavika. You are again trying to make it more and more complex. When you are trying to justify, you are actually trying to make it more and more complex. And in complex environment, you are going to need some of the messengers, some of the executioners, and so on and so forth. But what about existence of messenger RNA found in case of bacteria? This is a simple system, right? Had it not been essential, why would it exist in case of bacteria? That's the major question that you need to address. Okay, right. So, any logical reasoning why you should have the existence of messenger RNA? So, we are questioning the existence itself. It is present, that is for sure, that was actually deciphered by means of certain techniques. We'll talk about that as well. And the same logical reasoning was given because in case of eukaryotes, you have nucleus carrying the DNA, and the DNA never migrates from nucleus to cytoplasm. So how does the information gets relayed from a nucleus to cytoplasm, where the protein synthesis machinery actually exists? So that's what was the what you can say, the logic that was given behind finding the existence of messenger RNA. But that is much later. We need to first address this question that in case of prokaryotes, where there is no nucleus, where there is no distinction between nucleus and cytoplasm, where transcription and translation happen simultaneously, almost at the same point of time, why there is a requirement of mRNA, which is essentially just a copy of the gene, right? So any other reason? Bhavika, actually, partly you gave the answer last time itself. Yes? Do you remember what kind of answer you gave last time?
To control the gene expression, like to silence those genes that are not required.
Not exactly putting it in that way. Let me put it in slightly different way. Say, for example, you want to relay the information from the topmost official in the government panel. Say, example, it's the PM who wants to relay the information. What do you think? The PM will go to the clerk in Nagpur and will tell him that you need to do this directly, or it has a system to pass through, right? Why do you think why this system exists?
So that only that much information is relayed to the clerk, the amount of information clerk requires.
First and the foremost, there is a good level of regulation here, in the sense that when the PM is letting the information out, it is for sure be controlled well, in the sense that when the PM is letting the information out, if there are any flaws in the information, you can check for the flaws. Okay. And then the information ultimately will be relayed to the concerned person where the execution has to happen. Right? So you have a check here. And at every point of time when the information gets from the superior official to the person who wants to execute the information, every time it is getting relayed, it goes through a kind of a check. Do you agree?
Yes, sir.
Okay. So now at any point of time when you want to stop this information being executed, you can control it, correct?
Yes.
Okay. So is this a good way of having a control over the information execution? Because ultimately, just imagine this. Okay, you are spending some portion of energy in copying the information, fine. But ultimately, you are spending a lot of energy in synthesis of protein as well, correct? So if you can stop this information at this level itself, you will be saving the amount of energy, a lot of energy, what you are going to require for protein synthesis. Now, do you agree that compared to RNA synthesis, protein synthesis is going to require a large amount of energy?
Yes, sir.
Right? Two, when the information is coming from a very high authority, is it logical that that higher authority directly goes to the concerned executioner and gives that information? Because that will be a threat for the security of the higher authority as well, right? Did you get the point? Meaning, imagine PM going to the concerned clerk directly for telling him that you need to do this. Okay, if this happens over and over again, then don't you think that PM security is at a threat? Yes. Yes, exactly. So if you don't want that your official who holds all the information should be at threat, then you should relay the information through somebody who is intermediate, right? You can afford that the intermediate is getting the threats and all that. Okay, even if the intermediate is gone, the information still remains with the person who is actually bearing the information, correct? So imagine this situation by some or the other way. If the RNA gets degraded in between, it's fine, right? But what about you are directly relaying the information from gene to protein, and when the information is being relayed, the gene is directly getting affected because of the process in which the information is conveyed. Am I making sense? Yes.
Yes, sir.
So every time, imagine that you want to open the gene, you want to use it for entire amount of time till you want to go for protein synthesis. Okay. And in that time, it might happen that there are some kind of a chemical insults happening to the cell or something like that. The gene will directly get affected, right? However, imagine that the messenger RNA exists, and if there is some kind of a chemical insult, the messenger RNA will get affected, right? It will synthesize only a small lot of a faulty protein in case it still does. However, if the gene gets affected, the entire lot of proteins that you're going to synthesize are going to be affected, right? Am I making sense? So ultimately, are we arriving at some point of discussion where yes, exactly, Bhavika. This is the point that during RNA formation, it might get affected as well. But that will be for a shorter period of time compared to being utilized for protein synthesis. And two, then there is a gene correction machinery also that is working, which can correct it later on. Okay. But having a direct threat for the gene, every time it opens, there is a direct threat to the gene being affected by some or the other way, right? So during RNA synthesis, also there are chances, but this process is fairly, what you can say, mm, controlled compared to the protein synthesis mechanism. Every time you will want a protein, you will have to go back to the gene and find out, okay, I will be synthesizing as many copies you want, those many times you will have to read the information, right? Compared to that, in case of transcription, you have one copy of mRNA and several protein copies can be synthesized from this single copy, right? Compare it with gene. If you want to synthesize several copies of protein, you will have to use the gene those many times, as many times you want the protein copies to be prepared. So that much point of time, the gene will remain open and there is a chance of getting certain insults there. So will that be affordable to you? Obviously no, because that's where you're storing your information, right? So it's a kind of a locker system. You don't open the locker every time, correct? You have apart from your locker, you also have a savings bank account which you can operate, which you can use for doing the daily operations. You don't do your daily operations using a locker. So that's what is the difference between having the gene secure in the DNA and having some intermediary in between, which can, even if it gets some kind of a defect there, it's hardly going to cause any problem to the gene. So make sense? Yes. So one important way is you can have a higher level of regulatory control, and two is that you can protect your genetic information being affected during protein synthesis. So that is the reason why, first of all, now we have established the background that why there is a need of having an mRNA molecule at all. So we have already established this relationship now between the genes and proteins, and we have also now created a solid foundation of knowing that why there is a requirement of messenger RNA in between. Okay. Now, as both of you said, Sarvesh as well as Bhavika, as both of you said, considering this simple analogy, that in eukaryotic cells, the DNA resides inside the nucleus while the proteins are synthesized in cytoplasm, it was noted by, obviously, the most renowned Nobel laureate, Crick. In 1958, he was already a Nobel laureate, okay? And he noted this, that, okay, DNA resides in the nucleus and proteins are synthesized in the cytoplasm. So there has to be some connection that needs to be established between the nucleus and cytoplasm. He also noted something interesting. He noted that the ribosomes also contain RNA. And perhaps this RNA, that is the ribosomal RNA, is responsible for protein synthesis. Now, this was only a hypothesis, okay? He didn't actually give any proof of the concept. So he just put up the concept. He didn't give any proof for proving that, okay, the ribosomal RNA is responsible for protein synthesis. But this is logically a fitting kind of a concept, right? Because you have RNA existing in both areas, nucleus as well as in cytoplasm. Protein synthesis, as it happens in the cytoplasm, and as there has been a relationship established between gene and proteins, so there must be some information bearer who is carrying the information from the nucleus to the cytoplasm to execute that information for protein synthesis. Fine. Now, Crick noted this, that ribosomes contain RNA. So there was an obvious kind of a concept that he put forth, and that was ribosomal RNA are responsible for encoding the genetic information into protein information. So now we know that ribosomal RNAs are required for protein synthesis, but they are not bearing the information for protein synthesis. Is this fine enough? Yes? So now we go ahead from this point, right? See, a Nobel laureate is giving you a concept. So how will you disapprove that concept, right? You will have to have a solid way of putting it. Okay, you may be a Nobel laureate, but you may not be always correct, right? So there has to be a proper way in which you need to disapprove the concept that is given by a Nobel laureate, correct? So that was done by Francis Jacob and his two associates, Sidney Brenner and Matthew Michelson. He said Francis Jacob has had an alternative hypothesis. Okay. He said that the ribosomes are no doubt involved in protein synthesis, and they no doubt contain ribosomal RNA, but they translate an unstable RNA information bearers called as messengers, which carry the message from the DNA or the gene to be converted into protein. So they, in one way, Francis Jacob was trying to go against Crick and give another hypothesis, right? So for that, he, along with Sidney Brenner and Matthew Michelson, uh, arranged a kind of uh, an experimental system by the way of which he can, at one end he can disapprove the hypothesis what was given by Crick, and at the same point of time can prove the hypothesis what he is putting forth. Okay, so are we ready to look at what kind of an experiment was planned by Jacob's lab?
Yes, sir.
Should I describe you the experiment? Okay, the experiment is something like this. They used the T2 phage, infected E. coli, to switch the phage protein synthesis as their basis. Because as we know, that when the phage affects the host, or when the bacteriophage infects the host, then it actually hijacks the host protein synthesis machinery, right? So that has been already proven that the T2 phage will insert its genetic material into the host, and that genetic material will be utilized for the phage protein synthesis, right? Okay, are we good till this point of time? Yes. So the system is somewhat like this. If I put it like this, then a virus, or rather than saying a phage, let us consider that it is just a virus. Virus infects the host, and then what is happening is the hosts are now converted into viral protein factories. Is it okay if I say this? Right? Now, if the hypothesis that is put by Crick is correct, then for a viral protein synthesis, the host will have to synthesize new ribosomes, right? If Crick is correct, now if I put it like this, the Crick's hypothesis, if Crick is correct, then you will have to have a new ribosome synthesized, correct? Inside the host. So host will have to have new ribosomes. However, if Jacob's hypothesis is correct, then you don't need new ribosomes, no need of new ribosomes. Correct? So looking at this very arrangement, the experiment was designed. Now, what was done was that uninfected E. coli cells were first grown on a medium that contain heavy isotopes of nitrogen and carbon. The concept was that all E. coli, uninfected E. coli ribosomes will be heavy. Fine? Are you getting the point? So before the E. coli are infected, you are using them to be cultured on a medium that contains heavy nitrogen and heavy carbon. Okay. So obviously the uninfected E. coli's ribosomes will also be heavy ribosomes, right? So heavy ribosomes are containing E. coli are now being infected with the T2 phage. So now you have E. coli with heavy ribosomes, and now you infect it with the virus. Okay. As soon as you infect it with virus, you transfer these entire E. coli containing heavy ribosomes with the virus onto a medium that contains light nitrogen and carbon sources. That means normal nitrogen and carbon sources. So you transfer them into a light medium. Okay. Now you just check. What you will check now? Yes, if you have got the arrangement, what you will be checking? Can anybody tell me? Did you get the experiment? First of all, arrangement is pretty simple. Did you get the arrangement? Okay, yes or no? I just need to find out whether you got the point or not here. Okay, Bhavika says yes. What about others? Are you getting the arrangement clear? Okay, Sarvesh also says yes. Fine. So now what you will check? That's the question. What are you going to check? Yes, can anybody add in? Obviously, once I give you the answer or the question that they actually asked, then it will be very simple for you to imagine. But I now want to first give you the arrangement of the experiment. And what would you be asking to what question you would be asking if this is the arrangement? Let me find out how much you can have your imagination stretched.
Sir, if virus, if viral DNA is definitely making proteins, but if our rRNA is
Mhm,
responsible for the relay of information, then it will make new
mhm.
ribosomes, and the new ribosomes
Right,
would be light ones.
exactly, yes. And
So, if after the production of proteins, we still see the heavy ones present there, it means that it's not rRNA, it's
mhm,
some other mode of transfer of information other than rRNA, other than
r-
ribosome.
right, but you are also having your own hypothesis. One way you want to disapprove Crick hypothesis, fine, but you are also having your own hypothesis, right? Jacob's hypothesis is that ribosomes will translate an unstable RNA molecule, which is messenger, which is actually responsible for relaying the message from DNA to for protein synthesis, right? So, what if I have this kind of an arrangement that is on one hand going to disapprove Crick's hypothesis? That can be disapproved pretty easily, correct? Just by looking at whether whether you are getting new ribosomes formed after the E. coli has been infected with the virus. I can immediately disapprove Crick's hypothesis. Okay. Looking at if there are no new ribosomes formed, then the Crick's hypothesis is gone. It never holds ground. However, if there are new ribosomes being formed, can I still disapprove Crick's hypothesis? Because
No.
as the cell will grow, cell is going to synthesize new ribosomal RNA molecules as well, right? Are you getting my point? Because even ribosomes can undergo wear and tear, correct? So is there not a chance that although when you are transferring the infected E. coli cell into light medium, you still get some new ribosome synthesized with some old ribosomes as well? Then how will you disapprove Crick's hypothesis? Is this a valid question to put in now? Are you with me, Bhavika?
Yes, sir, I got your question.
Right. So, so I need to have another way of proving my hypothesis correct, right? So what was done is interesting. for any new phage RNA that is going to be synthesized, you have a P32 label there. Okay. So now what was happening is, the question that was asked was the radioactively labeled phage RNA getting associated with new or old ribosomes? Now, this is actually going to conclusively prove my hypothesis as well as disapprove the Crick's hypothesis. Are you getting my point? So now the system is well set. Although you're transferring it into a light medium, then you can have two setups. One, you will add P32 here and isolate all the newly synthesized RNA molecules, which will have the P32 label. Okay. And here you will analyze that whether there are any new ribosome synthesis taking place, which are light ones. Right? So in this manner, you can actually make out that what is actually happening, whether you are forming new ribosomes, and even if you are forming new ribosomes, are they translating the phage RNA? Okay. So what you can do here is that you can purify a tagged phage RNA, which will have a P32 label, and use that phage RNA to find out whether that is getting associated with old or new ribosomes. Make sense? Because the new ribosomes will be light and old ribosomes will be heavy. Is it complicating the matters for you? Yes? Or is it good enough? Okay, survey says no. So it's fine if you now find out one important stuff. And what will be that? You just need to assess now that what is happening. So if Crick's hypothesis is correct, the old ribosomes, which are heavy, with old RNA that are heavy and unlabeled, is there. Now you infect it with the virus and shift it into light medium and label the newly synthesized mRNA or any RNA molecule for that matter with P32. Fine. Now you find out that whether there are old RNA, that means heavy RNA associated with old ribosomes, or new phage RNA with P32 label associated with the light ribosomes. This is Crick's hypothesis, correct? So the Crick's hypothesis is only this area, while the messenger RNA hypothesis is given over here. So in Crick's hypothesis, what you should get? You should get the phage label, that is the P32 label with the new ribosomes. Is this fine? While this is the position of old ribosomes can be seen over here, and new ribosomes can be seen over here. So if the Crick's hypothesis is correct, then you should see association of P32 label with the new ribosomes. Fine. However, other way around, if the messenger hypothesis is correct, then you should see the old RNA containing ribosomes, which are heavy ribosomes, so heavy RNA with heavy ribosomes, still getting associated with another P32 labeled RNA molecule, which is newly synthesized RNA, that is the messenger RNA. So you should get the label with the heavy ribosomes, right? Make sense? Did you get the point? So you just need to see where is the peak position, whether you are getting the peak position corresponding to old ribosomes, or whether you are getting the p32 peak position corresponding to new ribosomes. If you get it with new ribosomes, Crick's hypothesis is correct. If you get it with old ribosomes, then Jacob's hypothesis is correct, that there exists a intermediate transient mRNA molecule, which you can call as messenger RNA, being relaying the information that was there in the DNA for protein synthesis. Are you having any doubts here? Or is the experiment clear?
Sir, I didn't get the last part where
Yes.
through density we are predicting.
Yes, obviously, density will be responsible for isolating old ribosomes with new ribosomes, right? New ribosomes being lighter, they will be less dense, and old ribosomes being heavier, they will be more dense. Correct?
Yes, sir, not that, but
Correct.
how are
The
we predicting?
p32 level you are.
I mean the shift in peaks like
Okay, okay. Go
old
ahead. Go ahead with.
ribosome. The peak that we get with old ribosome in both the graphs is before the peak that we are getting with new ribosomes. But there is a slight shift in the
No,
second graph.
look at the position. Look at the position that is given over here at in the axis that is written. This is bottom of the tube. So heavy ribosomes will be towards the bottom of the tube. Do you agree?
Yes, sir.
And light ribosomes will be towards the top of the tube. Do you agree?
Yes, sir.
Right. So old ribosomes are towards the bottom of the tube.
But in both the graphs,
What's the
still,
problem? Yes. It obviously depends upon how long do you continue the centrifugation. Meaning, if you are talking about the exact position of old ribosomes shown over here, with the exact position of old ribosome shown over here, it depends upon how long do you continue centrifugation, right? So how far does it travel? Whether it reaches to the bottom of the tube or whether it stays down the new ribosomes. That depends upon how long do you continue. And two, this is just not the scientific paper that I am referring to. This is included as a depiction to make it clear that what happens when you are looking at heavy ribosomes compared with old ribosomes. Does that make sense?
yes, sir. Yes, sir.
Okay, good. So ultimately now we have reached to the conclusion that there exists a messenger RNA molecule. So now we have established everything here to make our background pretty clear and pretty solid in terms of, first, that whether there is a need of an intermediate molecule. We have clarified all the doubts regarding that. Two, we have also proved that there exists an intermediate molecule, and that intermediate molecule is an RNA molecule different from the RNA that is present in the ribosomes. And hence, this new RNA molecule, which is utilized as a message bearer, carrying the message from the gene in order to be utilized for protein uh, uh, synthesis. So the message that is there in the gene is carried by this messenger RNA, and that message is read by the ribosomes in order to synthesize the proteins. So this completely proves and establish a solid base for now beginning the understanding about transcription reaction. So is this fine with everyone? Are you okay with all the explanations that we have searched and established till now? If there are any doubts regarding whatever we discussed today, then I'm there to answer. Or if uh, uh, you are not satisfied with the way that has been dealt, then you can also ask me questions so that the things become more clear in your minds. So if otherwise, if everything is fine, then I think that we are almost towards the end of the class. Uh, so you can leave. In case you want